In this video, you're going to learn about enzymes. We will cover today the fact that enzymes are proteins that act as a biological catalyst. We'll learn that a catalyst is anything that speeds up the rate of a chemical reaction, that a catalyst speeds up reactions by lowering the activation energy, and that cells use enzymes to speed up reactions necessary for life. For example, breaking down biomolecules such as protein in food into things like amino acids, or synthesizing new molecules from smaller parts. At this point, you should understand that enzymes are proteins that act as a biological catalyst. You should know what a catalyst is. You should know how a catalyst works, what activation energy is, and how cells use enzymes. The first thing you need to know is that an enzyme is a protein. So recall from our study of macromolecules that one of the functions of proteins was to speed up reactions. And now we see that enzymes are that particular protein that speed up reactions. They act as a biological catalyst, and a catalyst is anything, a molecule or anything else, that speeds up the rate of a chemical reaction. Enzymes are biological catalysts. Like we said before, a catalyst is a substance that speeds up the rate of a chemical reaction. But how do catalysts work? They work by lowering something called the activation energy. But before you can understand how catalysts work, you have to know what an activation energy is. Activation energy is the energy required to initiate just means to start, a reaction. So if you were using the example of our stair running from before, it's the energy you need to get to the top of all the stairs. How enzymes work is they lower the amount of energy you need. So again, in our stair example, it took less or a lower amount of energy to go up one flight of stairs than maybe it did to go up 2 flights. You would say this one has a lower activation energy because it requires less energy than going higher, which requires more energy and has a higher activation energy. On a graph for a reaction, you would see the activation energy is just the highest point on the graph. So it's wherever the graph goes up. And when a reaction has an enzyme, you would compare it to this red line in the graph. Okay? So this red line goes up not quite as high. It requires less energy, if this is the amount of energy required. If you compare that to the blue line, the blue line requires more energy. So in order to get up over this blue line, you needed to get more energy. This would be a reaction without an enzyme because it needs more energy to have happen. So if we go back to our graph, you could say this red graph is going to happen more frequently because it requires less energy. So in the case of an enzyme, it makes it so a reaction needs less energy to happen. Another example I could say is if I told you to go drop something off in the Freshman Academy office, that would not require much energy. But maybe if I asked you to go to the 3rd floor office, it would require more energy. Because it takes less energy, enzymes also make it possible for a reaction to happen more often. Think. How could you compare this to the stair challenge we did at the start of the period? What is the connection between how easy something is and how many times you can do it? Write this response in your notes. Okay, so now you should know what an enzyme is, what it does, and now we're going to talk about how it does what it does. So how do enzymes lower activation energy? Before we can answer that question, there are a couple of things we have to know. The first thing is how to look at a chemical reaction. There are 2 sides of every chemical reaction, and in the middle of those 2 sides is always a big arrow. On the left side is something called a substrate. The substrate is at the beginning of the reaction. It's called the reactant. and it is what is reacting. Substrates or reactants are always on the left side of a chemical reaction. So if we use the example below, water, salt, yeast, and flour give you bread, the substrate or reactants are the water, salt, yeast, and flour. They are what you are reacting or what you're combining at the start of the experiment. Then you have products. Products are always found on the right side. Products are what you produce. So they're what you end up with at the end of a reaction. In the example, if you take flour, water, salt, and yeast and you combine them, what you end up with is bread. So that is the product. Now it's time to check yourself to make sure you understand everything you've seen. Answer the following 3 questions that are found in your notes. Now, pause the video and try this one. Which colored line, red or yellow, represents a reaction with an enzyme in it? Write the answer in your notes. Finally, find this last check yourself. In the reactions below, hydrogen plus oxygen gives you water, and carbon plus oxygen gives you carbon dioxide. Circle the reactants and double underline the products in your notes. Alright, back to our question. How are enzymes paired with the right substrate? So if substrate's what's reacting, how do enzymes pair with the right one? And how do they speed up reactions? Every enzyme has an active site. The active site is this area right here, and the shape of the active site determines what substrate can bind. If a substrate doesn't fit perfectly, to emphasize perfectly, no reaction is going to take place. So in this case, you can see that this shape of the substrate fits perfectly in the active site here, so those two can partner up. Many times people refer to this as a lock and key model. So enzymes and substrates are like a lock and key. Why does this make sense? Because if the enzyme is the lock, there's exactly one key that can fit inside of it. It doesn't matter if something almost fits. If it fits perfectly inside, it will unlock the door in this case, speed up the reaction. If it doesn't fit perfectly, even if it's really close, nothing good's going to happen. Try this. Label. Determine what each letter is pointing to from the vocab. So what's A pointing to, what's B pointing to, and what's C pointing to. Record your answer in your notes. Looking here, you should have said A is pointing to the substrate, B is pointing to the active site, and C is pointing to the enzyme. The last thing that's important to realize is enzymes are really important. They help us get energy for food. They're enzymes that break down carbohydrates, proteins, fats, and pretty much everything in your body. And what you'll notice if you look at this chart is, again, looking at this chart, maltase, lactase, cellulase all end in ASE. Why is that important? Because if you're ever taking a test or quiz and you see something that ends in the letters ASE, it is probably an enzyme. You've completed the video on enzymes. Go back, watch any part of this video you need to watch again, make sure you completely fill in your notes, and ask questions if you have any. Enzymes almost always end in the letters ASE. Catalase, amylase, lactase,